I'm Dean Kereiakes, and I'm the medical director for the Christ Hospital Heart and Vascular Center, the Christ Hospital Research Institute, and I'm a professor of clinical medicine at The Ohio State University. Yeah, no, CAD I and CAD II are small trials. They weren't adequately powered, and there was not an effort made at comparison. In lieu of a randomized comparison, we constructed Disrupt CAD III very carefully to use performance goals, to create performance goals from the prior FDA IDE trial for orbital atherectomy, which is Orbit II. And using that trial with roughly a similar number of patients, we were able to try to use the same population, same definition, same primary endpoints, so that we could do the best possible short of an actual head-to-head randomization in an adequately powered trial to, as you say, prove safety and effectiveness. So the safety and effectiveness had not been proven at the level, a bar set by the US FDA, and CAD III allows us to do that. This is a non-randomized single-arm study where we created performance goals based on the precedent IDE trial for the approval of orbital atherectomy, which was called the Orbit II trial. Orbit II was also a non-randomized single-arm study where they created performance goals based on prior trials of heavily calcified vessels. I think what we did is probably more accurate in that we actually tried to establish the same patient population, the same definitions, and the same primary endpoints. So, for example, we identified patients that were heavily calcified. We had to get into our study, either radiopacities on both sides of the vessel without contrast in the lumen angiographically, at least 15 millimeters on both sides of the vessel by angiography. Or you could have, if you did imaging, at least a 270-degree angle or arc of calcium. So these are pretty high bar for patient selection, radiographic or imaging. Second, we identified the same primary endpoint for safety. That was freedom from MACE at 30 days. And that was cardiac death, MI, target vessel revascularization. And we identified the same primary effectiveness endpoint, which was successful placement of a stent with a less than 50% residual stenosis and no in-hospital MACE. Now, that's an archaic definition. We understand the less than 50%, but that was the definition used in the Orbit II trial. So, as a sensitivity analysis, we pre-specified that we would also test the same definition, but using a less than equal to 30% residual, which frankly has been incorporated in most FDA IDE studies of the last 5 years. So, interestingly enough, as a sneak preview, there was absolutely no difference. One of them is 92.4%, the other is 92.2%, absolutely no difference. So it didn't matter whether we used less than 50 to be fully consistent with Orbit II, or if we used less than equal to 30% to be more contemporary. Those are the two primary endpoints. We then enrolled the patients and used also core lab, CKMB. This is very important, and I think you're going to see more and more trials doing this. When you have a precedent trial that you're comparing to, and they used only CKMB as a biomarker for the diagnosis of periprocedural MI, and using a greater than 3-fold elevation. Now, some would be critical that that's not as important as a 5-fold or 10-fold using the new SKY definitions. We get that. It doesn't make our data look less attractive. In fact, it makes our data look significantly more attractive very frankly, from a MACE standpoint. In fact, the myocardial infarction rate, if we use the SCAI definition, is 2.8%. But that said, there are many centers, particularly in the United States, that don't use CK&B. They don't do it. So we actually core lab those centers. Rather than trying to adopt a definition with troponin, which we've now learned is very hazardous as of presentation of another pivotal trial at TCT, very frankly, where troponin aberrated their target lesion failure estimate and made it almost 2 times what they had powered the study to be. We powered our study based on endpoints that were derived from the Orbit 2 trial and set performance goals with FDA guidance based on those endpoints. And we wanted to use the very same definition for paraprocedural MI to make these almost the equivalent of a head-on comparison. So that's how we constructed the trial. And I'm happy to tell you how it worked out. Well, first, number one, the primary safety endpoint, freedom of MACE at 30 days. That was achieved in 92.2%, and the lower confidence bound of that was 89.9, which was far above our performance goal of 84.4. So far and away, we hit the primary safety endpoint. And the primary effectiveness endpoint of procedural success, same way. The lower confidence bound of the 92.4% far exceeded the performance goal that we had established of 83.4, which was derived from the procedure success rate in Orbit 2. And the primary effectiveness endpoint was met. As far as perforation, abrupt closure, no reflow, following IVL, the answer is 0. With a heavily calcified vessels, and I want to stress that point in a moment. As far as the perforation, there's one microperf after stenting, which is IVL alone and then after stent, but that didn't require pericardial. There was no pericardial effusion, no MACE to 30 days. Patient went home the next day. I mean, basically sealed with a prolonged inflation. Is very different than many of the perforations that we see following ablative technologies like rotational orbital atherectomy. The other, really, I called it jaw-dropping observation, was related to the 100-patient OCT imaging substudy that was the lead author on that for TCT was Richard Schlafman's. Despite the fact that our average calcified length was 48 millimeters compared to Orbit 2 of 28.6, dramatically different, much more heavily calcified vessels. Lesion length, 26 millimeters compared to 18.9 in Orbit 2. The average arc of calcium in our study was 293 degrees. You know that anything above 180 is a significant risk predictor. That's the calcium volume index scoring system that's now been embraced by the SCAI consensus paper, just published in CCI by Riley, Henry, Jaffer et al. And that says an arc greater than 180 is the number one predictor. You get 2 points for that out of 4 total points in the scoring system. So our average arc was 293, and the average thickness of calcium at the site of maximum calcium was almost a millimeter. It was 0.96. The CVI calcium scoring risk index uses 0.5. So we're almost 2x that. So this is, I think, by far the most heavily calcified group of patients ever enrolled into a cohort for an IDE study. And despite that, the average stent expansion by OCT was 102%. And the minimum stent area anywhere in the target vessels was 6.5 millimeters squared. So those parameters, when you achieve those results in that population of patients, it's truly striking and should predict excellent long-term results, meaning low incidence of restenosis, low incidence of thrombosis, because when you get stent expansion over 90% and MSAs in that area of over 6 millimeters squared, that's really striking to see that. So I think that was one of the real eye-openers, so to speak, of the results that we're able to achieve. And the other thing was the ease of use. We actually had every center, 47 international centers in 4 countries, okay? US, UK, Germany, France. And so everybody got their first case. We took those 47 and we analyzed them compared to the 384 in the ITT population. Now, think about learning a new novel technology. Sometimes it can be challenging. I was a proctor in the 90s for rotational atherectomy. I used to go center to center, training people how to do it, okay? Parameters of success, including device crossing success, freedom from MACE at 30 days, and procedure success in those 47 was no different than the 384. How can there be no learning curve? Well, the answer is it's a balloon. If you take a novel technology and you package it in the most primitive delivery system that any of us as interventionists use, dates to the days of Andreas Grunzig, of putting a balloon into an artery. It's a balloon. So it amazingly simplifies the procedure. The only issue is getting it in place. And if you can get it to the target, which we did 96% of the time, it's pretty darn good. I think the crossing failure rate for this device was not as high as what we frankly saw with the Absorb stent, when I was the principal investigator for that trial too, Absorb 3. So this is relatively higher profile. It's more high profile than an NC balloon. But if you can get it to the target, it's almost a no-brainer. This has, some of us argue there really is not a learning curve because everybody's used balloons for years. I think it's ingenious. They take the technology that's been used to treat kidney stones and fractured kidney stones with ultrasonic pressure waves for 30 years. And they have packaged it into a fluid-filled balloon for delivery into the vascular system. And it works. We now know by fracturing calcium in a very unique way. It's circumferential and longitudinal in multiple planes. And there's nothing else, no other technology that does that. The only way that the ablative technologies, whether it's rotational, orbital, or laser, can access deep calcium is through extreme wire bias. Think about it. They track a wire. And if you have a very deep, we say medial but deep calcium, you will get a rut or a trough. It's very eccentric. This doesn't care where the calcium is. It's circumferential and transmural. So it gives you a much more predictable, reliable, and safe modification of calcium throughout the vessel wall. This allows you to increase the transmural compliance of the vessel, much more compliant. It markedly reduces fibroelastic recoil. So the MLD, minimum lumen diameter, you get even after the IVL balloon alone is truly remarkable. Almost like a stent-like result, despite the fact that the maximum inflation pressure in the IVL balloon was only 6 atmospheres for the entire study. So where else can you size a balloon 1-to-1 with the RVD, reference vessel diameter, go only to 6 atmospheres in arteries that have the characteristics that we describe and get full balloon inflation? It simply doesn't happen unless you modify the calcium in a circumferential transmural way. So that's the biggest advantage of the technology, I think, is the safety, ease of use, and then its effectiveness with respect to deep calcium. IVL is safe, it's effective in modifying calcium and optimizing stent deployment in heavily calcified coronary arteries. It's predictable and reliable and has remarkable ease of use, so that the learning curve is abbreviated to the point of being almost non-existent. The other thing that we saw in this trial that was pretty interesting is anybody who's done these procedures, even in the periphery, sometimes you'll see spikes on the EKG. And when you do it in the coronaries, not only will you see spikes, but sometimes you'll see a PVC called shock topic, or suddenly it looks like there's a pacemaker. And particularly patients by multivariate analysis that have heart rates of either less than 60, less than or equal to 65, are more likely to have their heart rhythm usurped by, taken over by, the IVL for the 10 beats that you do. And then as you do the procedure, you inflate the balloon to 4 atmospheres, very low pressure. You deliver 10 pulses. Then you go to 6 atmospheres, which is a nominal balloon inflation pressure. If you don't get full inflation, you take the balloon down, reperfuse the patient, repeat the cycle. Eventually, you're going to get full balloon inflation at only 6 atmospheres of pressure, which is the nominal pressure. But during the 10 pulses, it's 1 per second, each a millisecond, that transmits at least 50 atmospheres of pressure instantaneously. What happens there is electromechanical transduction. So suddenly, this paces the heart. Everybody wants to know, can you cause RNT? Can you cause VTVF? The answer is never been seen. There's not enough energy generated to do that, very frankly. And it's a benign curiosity observation. We had 41% of the patients in the trial. I mean, that's a large number. We originally set out and designed a trial to have a 20 or 30 patient sub-study of this, specifically at the request of the FDA. And now we've got a lot more patients, and it's benign. Patients who have IVL-induced ventricular capture more frequently drop their blood pressure, systolic blood pressure, during the procedure than those without capture. But the magnitude of drop in systolic blood pressure is no different between these groups. So, and for 10 seconds. So if I had somebody who was really marginal, high 90s, kind of a poor candidate, maybe impaired LV function, I might give them a little volume if they're dry, maybe give them a little niacin to tighten them up a bit. But for 10 seconds, it's not really a big deal. Then you go to 6 atmospheres for a couple seconds and suck it down. I mean, your entire treatment cycle is 15 seconds or less. You see, so I mean, it's not a big deal, and sometimes you can pace through it, but it was no clinical consequence. There's frankly never been a time in our history as interventionists where vascular calcification is a bigger problem. Between the aging population that we're working on, the most rapidly growing segment of the population in the United States is over 80. The prevalence of diabetes, the prevalence of chronic kidney disease, GFRs under 60, all of these combine to create more heavily calcified vessels than ever before. And never in our history as interventionists has there been a greater need for large bore catheter access also. Not only is there calcium in the coronaries, up to 25% have moderate to severe calcium. In the iliac and femorals, up to 50% have moderate to severe calcification. And with the miniaturization of technology, everybody on both sides of the ocean have had an explosion in EVAR, TVAR, TAVR, mechanical cardiac support devices, all of which define transfemoral access as the access of choice and associated with the lowest incidence of complications. Nothing simpler and easier than a perc access, per close, vascular closure device, in and out. I mean, they literally can go home the same day or the next day, as opposed to a cut down, an alternative vascular access. And that is much more resource consumptive as well. So this actually fits very nicely in the algorithm for care for mechanical cardiac support in our center. In fact, you can tuck the cost of the device into the reimbursement for the mechanical cardiac support device. I think it is costly, but at what cost is alternative access? What cost is procedural failure or inability to do the procedure, very frankly? And I think those costs are not inconsequential. And all of us that are interventionists, think of those terms. So I think the cost is always a consideration for any new technology. I'm very hopeful there'll be incremental reimbursement in the coronary space because it is a breakthrough novel technology. It's been defined as such by the FDA, and I think this trial, Disrupt CAD III, proves that point. I think immediately there were patients that were excluded from the trial, patients with osteobifurcation disease, even larger vessels, and patients that had to have interaction of technologies like an ablative technology plus IVL. They're not mutually exclusive. For example, presently I would use a 125 or 1.5 burr if I couldn't cross with anything. And there are times because of superficial calcium, you cannot cross with any device, doesn't matter what you use, guide extension, et cetera. And in those technologies now called rototripsy, rotoshock, you get the ability to access and place the IVL balloon. And they're really complementary. One of them will access and ablate the superficial calcium that will allow you to put the IVL technology in place, which then can modify all the deep calcium in the vessel as well. But the only way you can modify that with the ablative technology that's wire guided is to have extreme wire bias, and then you do it through an isolated trough or rut and not in a circumferential longitudinal fashion as you would with IVL, which is much more effective in altering the compliance characteristics of the vessel and eliminating fibroelastic recoil, and then mitigating the need for high pressure balloon inflation, which is really key. These very high pressure balloons that you have OUS up to 30 plus atmospheres. Sometimes we do that here in the US too, but this is, boy, when they burst, it's not a pleasant phenomenon usually. This really tears the artery. It's intuitively unattractive to have that kind of barotrauma in a vessel. Now you don't have to do that. The only time I go to high pressure in an IVL case is to post-dilate the stent to further optimize the stent. And it's really an amazing sequence. I mean, it's really facilitates stent optimization.